Hello, everyone. Good morning from whichever part of the continent you're joining us from. Welcome to today's webinar session on FCM, a systems approach to FCM management, presented by Bioline Agrosciences and specifically Bioline Academy, which is the training arm of the Bioline Agrosciences, formerly Dudutech. Welcome, everyone. In today's session, we'd want to talk more about the little things that add up to help us manage FCM and specifically for the export guys where FCM is a pain in the wrong places. I think without any much ado, I'd like to introduce our presenters for the day. Our first presenter would be Dr. Megan Malkahi from River Biosciences. We are partnering with River Biosciences in this particular webinar, together with the Kenya Plant Health Inspectorate Service, KEFIS. So Megan will be the key speaker for the day. She's going to take us through FCM identification, taxonomy, biology, and also the systems approach to management. For exporters, we know that FCM is a pain in the wrong places, and we got a regulator in the house from a Kenyan perspective, and that is Kenya Plant Health Inspectorate Service, KEFIS. And we've got Dr. Mary Gwantai, who is going to talk to us about the regulator's perspective to FCM management. So without any much ado, I'd like to welcome Megan. Megan, kindly take it away.
Thank you so much, John. I'm just going to share my slides. If you just bear with me for a little bit while it loads. Okay. Can everybody see just fine? John, it's working?
Yes, it is.
Okay, great. Sorry, I will just put
presenter
it in
mode.
Presenter mode. There we go. Okay, hello everybody. My name, as John indicated, is Megan Malkai. I work for the River Bioscience and Exit Group. And today, obviously, we'll be talking about false codling moth, more specifically a systems approach to the management of the moth, and then going a little bit into the background of false codling moth, how to identify it, how to monitor for it effectively, etc. So obviously the background of this is the huge difficulties being faced by the rose industry with large numbers of roses being culled in order to look for potential infestations of FCM. So just to get started, I thought I'd put up a little bit of an agenda here because it's quite a long presentation and hopefully we can keep it nice and orderly so nobody gets lost in actual discussion about the FCM. So first we're going to just go into an introduction on River Bioscience, why am I here presenting to you about FCM today? So a little bit about our history and especially because I think it can show you that there is sort of a light at the end of the tunnel. We were able to get FCM more under control in South Africa, and we've got a large history with that. And so there is positivity that the rose industry can achieve the same. Then we're going to go into the FCM itself. What is it and why is it so problematic? While we're going through this, we'll also do some moth ID, look at the life cycle as well as the larval ID. Then I'm going to discuss setting up a holistic area-wide pest management program. So what are some key lessons we can learn from citrus and other industries in South Africa? And then what are the key aspects of FCM management in roses? So specifically looking at scouting and sanitation, because these are really the cornerstones of good FCM management. And then finally, looking at pest management tools. So the use of mating, disruption, foliar applications, and biocontrol together in an integrated program to control the pests. Okay. So River Bioscience, is focused on commercializing and distributing sustainable crop protection technology. And our goal is to focus on key pests using holistic and accurate programs. So when we say holistic, it's looking at the entire life cycle of the pest. And this is our approach to FCM, not just focusing on one aspect of it, but the entire life cycle along with the cropping system that it's in. And our aim is really to support the competitiveness and sustainability of high value crops. This is really, goes into why we're assisting with the rose market, because we want South African and African in general crops to be very successful on the world stage. So how River Bioscience fits into the South African fruit industry is that we actually fall under Fruit South Africa, and more
Sorry
specifically.
to interrupt, Megan. Would you kindly turn on your camera as
my
you continue?
camera.
Sorry about that. Yeah, or it's not working.
Sorry, I will try, see if this is working. I'm sorry, it's not allowing me to do it on the web browser.
Not a problem, not a problem. Carry on.
Yeah, sorry, it's got the little thing there. So apologies for that. Perhaps when we are doing the question and answer session, then I will join using a different method to try and use the camera.
Sure.
Okay.
Just go on.
You can still see the slides?
Yes,
John,
yes,
can you still see
clearly.
the slides? Okay, great.
Sure.
So as I was saying, River Bioscience falls under the Fruit South Africa umbrella. Specifically, we are part of the Citrus Growers Association. So we're really grower owned, which is why we're very much focused on grower problems in our continent. Making sure that our products are available on international markets. Like I said, as a grower owned company. Okay, this means that we do have quite a bit of history within South Africa with regards to pests that are problematic on high value crops. And one of these pests very specifically is the false codling moth. So we have experience in area-wide management over 10 years, as well as over 5 years specifically of mating disruption, and over 20 years of FCM control with foliar applications, as well as a vast monitoring network. So, we really do understand how this pest works in South Africa, and then how best to do a program in order to control them. So, when we come into the rose industry to give advice, it's really coming from that experience. So without going further into that, I'm going to jump straight into our enemy number one, and that is the false codling moth. So this is the moth over here. The species name for the false codling moth is Thaumatotibia leucotrita. And on this picture, I'm just going to talk a little bit on how to identify the adult. So the false codling moth is quite a plain brown, sort of drabby, speckled looking moth. There's not much that really sets it apart in terms of, flamboyant markings or very bright coloring. So what you want to look for is you want to look for the little tufts that you can get on false codling moth. These can be found in other tortoise species as well. So just keep an eye on your traps that you're not catching some other tortoise species. For the false codling moth, you do get this sort of speckled aspect to the body of the moth with the scales. And then something that can help to set it apart from some of the other Tortricidae moths that you might catch in traps is that false codling moth has this little C shape over here. So it's got these dark markings on the wings that form this kind of C shape on both sides of the body. And then typically when it does land in traps, it'll typically land with the wings closed. Not always, but it does often do that. Something else to have a look for is that you can't really see as well in this photo, but there's fringes on the edges of the wings. If you do ever have some doubt about, what moth you're actually catching in your traps, you can always send photos, perhaps to the Bioline chaps, who can then forward the photos to me if you want some input on the identification. But with our monitoring traps that we're using nowadays with the pheromones, the likelihood of you catching a lot of other species is very low. So typically in those traps, you will catch false codling moth. So this is just showing the life cycle of the pest over here. So you can see it goes moth to eggs, then the larvae will start to bore into the crop. In roses, will bore into the bud at the base of the rose. And then it will complete its life cycle within the crop up to 5 larval stages. And eventually it will exit the crop, spin down on a bit of silk, or if the crop falls to the ground, then it will also reach the ground. Then what happens is the larvae exit the fruit or the crop, and then it will pupate in the soil. So something that makes FCM so difficult to manage is, firstly, FCM is a native pest in sub-Saharan Africa. This means that it is very difficult to get control of the pest in a very long-lasting way. You always have to sort of employ management techniques. The reason being is that this pest attacks up to 80 different host species. So it's a polyphagous pest. It attacks multiple different types of crops. So even if you get good control in your greenhouses or in your fields or in your orchards, there's always that risk that you can get reinfestation from other host crops in the area. So other host crops could include citrus, for example. In South Africa, it can have table grapes, stone fruit, pomegranates is a very big one, but also some native plants. I'm not as sure in Kenya or Ethiopia, but in South Africa, for instance, we have a plant called kai apples, which is very problematic, and another one called boer beans. So there's multiple different crops, both native and cropping plants that can act as a host to this pest. So having a look at what your cropping system and what your bush in the area of your greenhouses, what types of hosts could be in there, and then just keep that as an option for your sanitation practices, maybe removing those hosts. Another issue with FCM is that there's no diapause, so that means it doesn't go into a sort of hibernation. This means that it will just tick on slowly during the winter, and then there's the opportunity for the numbers to get very high very quickly when temperatures heat up, especially in the more climates that typically have warmer winters as well. This also means that there's overlapping generations, so it's quite difficult to identify when do you need to start doing your management practices, and this means that you have to have very good monitoring processes. There's also strict chemical regulations, especially for food crops, on which types of chemicals that you can use, with a lot of active ingredients being removed from the market. So not only do you have to control it better, but you've got less things to control it with. And this is why integrated pest management is so important. And then finally, the phytosanitary status. So what we mean by phytosanitary status is that other countries don't want false codling moth to enter their markets. And this is because false codling moth is such a big pest on multiple different crops. So these countries want to keep FCM out of their borders so that it doesn't become a problem for them. So out of our phytosanitary markets, there's, the United States, South Korea, China, and probably more important for you guys is the EU. So this is why they actually destroy or monitor the crops coming into the country to look for infestation. And if they find infestation, then there's the opportunity that they will reject that import coming into their countries, and then potentially stop you from exporting in the future. So this is why the control of this pest is so important. There's almost a zero threshold for the infestations. So before I get into how do we manage this phytosanitary pest, I just thought I would go into the morphology. So how do we actually identify it? So I already spoke about the larval ID, but now I want to talk about, I already spoke about the adult ID, that sort of speckled body with the little black C on the wing. Now I'm going to talk about the larval ID. So over here, I've just got a picture of a generalized larvae, but this is what the FCM larvae looks like over here. Now, the things that you want to have a look at for those generalized areas is the head, the thorax, and the abdomen. So something that's important for identifying FCM is having a look at the spiracles, and you can see that the spiracles are on the abdomen area as well as on the thorax area over here. So just in terms of key aspects for identification of FCM, the first aspect is the posterior of the FCM larvae. So the important part here is something called the anal comb. It's like a little comb that's on the back of the larvae. So FCM have this anal comb. It's sort of like a 5 to 7 pronged little comb. And then some other larvae that you might get in the areas don't have this anal comb. So this is just an example of carob moth, for instance, so you can see how it differs from the FCM. So one of the first things to look at if you're trying to identify an FCM larvae is, does it have an anal comb or not? Something else that we want to have a look at is the spiracles on the anterior over here, and then the posterior of the moth. So in the anterior, you've got this section over here. This is called the L1 section. So this is the first section of the larval body. Then what you're looking at here is the spiracle, and then this section over here with the setae, or little hairs of the moth. So in FCM, the spiracle has setae that are lying below it. In other species, sometimes you can find this L1 region over here on the side or above the spiracle. But for FCM, this section is below the spiracle, and there's typically 3, potentially 4 of these little setae coming out of that L1 section. On this posterior region over here, you've got another spiracle over here on the A8 region, or the abdominal region number 8. And then you've got a little setae over here as well. And typically this setae is on like a little raised bump. In other species, sometimes you get a ring around the setae, or sometimes the setae is in a different location. But for FCM, the setae is, or the little hair is right next to the spiracle, and it's on a raised little bump. And then obviously you can see that anal comb at the back there, and then you can see how the setae is arranged over here on this region. But the most important aspects are that anal comb and then this L1 region. Those are the easiest ones to tell the difference between the larvae. So you typically need to, if you can see, look at these under the microscope. So if you are having difficulties, or you're concerned that maybe you have a different species and not FCM, then I would encourage you to reach out to Duditech and the Bioline guys. So this is just another aspect of that anal comb that you can see there. Okay, now that we have discussed the identification of the larvae and the adults, I wanted to go into the principles of a holistic area-wide integrated pest management program. And in this section, we're going to learn a little bit of the lessons from citrus and how we do management in citrus in South Africa. So this is sort of our philosophy on area-wide integrated pest management. This involves the multiple tactics or multiple different tools that are employed over wide geographic areas to achieve pest population and reduce phytosanitary risks. So you can see here that I say population suppression, not population control, because as I mentioned already, it's virtually impossible to get full control over FCM. That's because there's always that risk of reinfestation. So what we want to do is suppress the populations within our orchards, and then make sure every season that you're doing the correct things, because you will never get full control and you can never take your foot off the pedal, for instance. So one of the things to achieve good holistic area-wide integrated pest management is to follow these principles over here. So the first one is to understand the risk on your farm. And to do this, you need to know the pest quite well, but you also need to know your farm. So know which areas are most at risk of having infestation. Then you want to establish a foundation program. So that means taking the most basic steps every time and making sure you employ those correctly. And I will talk about this later, but these basic steps include sanitation and monitoring. You know, build a good base for your management program. And that's foundation will help you get better control over the season. Next, you want to establish site thresholds using monitoring networks. So obviously for FCM, the threshold for larval management is very low, you know, almost zero, because you want no larvae going in with your crops when you export them. But the populations of the adults will typically not always be zero. And the best way to make sure that you're monitoring, you're managing them correctly, is to understand what the population is doing, and knowing, as I said already, which areas are most at risk, which areas are you constantly having difficulties with. Then you want to allocate your resources wisely. So you want to select products very carefully, select products that you have confidence in, that you have technical support for, and make sure that you stick to the labels, especially when you're using softer methods or biological control methods, because these methods do work as well as synthetic chemistries, but you have to use them correctly. So make sure that you're adhering to the labels. I'm not as familiar with the registration processes in Ethiopia and Kenya. I'm sure Mary will speak about that later, but make sure that you're using products as they're recommended and, as they're registered. And then finally, set realistic expectations. So you need to realize that there are no silver bullets. There's not just one thing that you can do to help get control or management of FCM. You have to use an integrated approach. You have to use multiple tactics together in a way that will give you that FCM management system. So I'm just going to give you some of these lessons from citrus. And the first thing that I mentioned earlier was understand your risk on farm. So, how does your different varieties or cultivars affect your pest management? Because some of these things are more susceptible. For roses, I'm not as familiar, but this is just an example from citrus. So you can see that typically the navels are experiencing some of the highest pest pressure. And in the past, lemons were quite low, but as we started to get more and more suppression of FCM in some of the other susceptible varieties, you can see that the moths start going into the lemons, because lemons are typically less well managed, because it's not as susceptible to FCM. So just because you think something is less susceptible doesn't mean that you shouldn't monitor for it to see, is that population changing over time? Next, I spoke about establishing a foundation program. So this is quite a busy slide, and I'm not going to go into the huge detail of it, but this is basically how a system management looks for FCM and citrus. And you can see there's multiple overlaid tactics, and the base is here. This is the foundation. And the foundation is that sanitation, picking up of out-of-season fruit, and then removing infested fruit from the trees in the season as well. And then on that base, you have your monitoring, you have your area-wide approach, and you have your corrective sprays. So the reason why we're looking at a systems approach is because every link in the chain of FCM management has an impact on how successful you're going to be when you're trying to export your fruit. And this can be seen quite clearly over here, where you can see there's a statistically significant relationship on what happens with FCM management in the full season and what happens in the last 4 weeks before harvest. Obviously, for roses, the harvesting and the managing the timelines are much shorter. But what you need to think of this as is you need to start early with your management, and you need to be on top of your management. Otherwise, by the time you're harvesting, you're going to have a problem. And it's too late to manage something by the time you're harvesting. Thanks, Sean. Sorry, I just had someone come into my office. Okay. And then finally, there's a statistical significant link between FCM infestation in the last 4 weeks before harvest and then ability to deliver insect-free crops to the pack house. So what this means is that, you want to get your early season management correct. You want to manage it throughout the growing period. You want to do everything that you can to get it to the pack house free of infestation, so that it's easier for your sanitation teams and your pack house laborers to then pick out those infested flowers and then deliver infestation-free plants in the export. I hope that makes sense to everybody. But this is what I was saying about every link in the system is important. So you can think of this as your management chain. And if you break one of these chains early on, or if you don't do it correctly, that means that you're going to have a problem by the time you export. So you want to do everything in your management system as correctly as possible. And that starts all the way from orchard monitoring and orchard sanitation and pre-harvest controls to shipping regimes by making sure that you're doing culling during the harvesting process and that you are doing your inspections at the pack house. So this looks like this. Once you sanitize, you reduce the number of insects. Once you use registered controls, you reduce the number of insects. Then through monitoring and doing appropriate threshold applications of foliar insecticides, for example, you reduce the insects, and so on and so forth through every type of control method until you're left with 0 insects. So it's that's what we mean by a systems approach, you know, looking at the system as a total and doing individual management tools throughout that process. So for River Bioscience, our management system is a 20-year investment, you know, looking at how do we meet food safety, world-class quality with our fruit, avoid retailer restrictions, especially with chemicals, and then achieve those phytosanitary measures. So how we've done this is to try and control FCM at every stage of the life cycle using different methods, you know, looking at area-wide methods as well as biologicals, as well as foliar applications. And this is sort of how it looks like when you put it together on the life cycle of FCM. So this is what I meant by a holistic management program, and then making sure that you're starting out at the beginning of the season with your basic steps, you know, the monitoring and the sanitation. So this is what we would like to repeat for roses. So taking those lessons that we've had in South Africa with controlling this pest and applying it with your roses. So this is where I come into why the basics are so important. I know I've already mentioned this a couple of times, but really it is the key to FCM management. And the first key to FCM management is your monitoring practices. So I have had a discussion with a couple of different people, and it seems like there was some variability in how people are monitoring the FCM in roses. But really the most recommended way of actually achieving good monitoring standards is to use your yellow delta traps with the correct FCM sex pheromone as a lure placed inside those delta traps with a sticky pad. Then you want to place approximately 1 trap per every 2 hectares. In citrus, we do 1 per every 4 hectares, but the management requirements for roses is much more intensive because the growing period is shorter. So the recommendation would be about 1 trap per 2 hectares. You want to make sure that the traps are spaced correctly. So you want to make sure that they're at least 150 to 200 meters apart. This is because if the traps are too close with each other, they will interfere with each other, and then you won't be able to catch the correct number of moths, and it won't give you a good indication of what the population is doing in the greenhouses or in your rose fields. And then finally, you really want to consider the air movement in the greenhouse and apply the trap accordingly. So what you want is to make sure that air is moving through the trap so that the pheromone can dispense towards the moths, and then the moths will come and they'll move along that pheromone until they reach the trap and then get caught in the trap. You can then do your counts in that trap to see what is the population doing, and then monitor those counts over time. So see when is the population decreasing, when is it increasing, and that can help you identify when you need to do applications of various management tools. So this is just having a little bit of a look at trap placement. So typically the traps are hung by a wire and they look like this. This is the yellow delta trap. It comes flat packed and then you build it. So you need to make sure that the wing flaps are in the correct position so that it maintains that triangle shape. You need to make sure you've got your sticky pad in there and that you've got your pheromone basket or your pheromone holder that is loaded with the pheromone. Typically in orchards, we want to place the traps about 1.8 to 2 meters high. In roses, I think it would be more between 1.5 to 1.8 meters. And you can hang them either from the structures in the greenhouse or on poles. So this is an example of a pole over here, and that's more than adequate to hang them. In greenhouses, what you want to understand really is how your air is moving. So in the orchard, obviously, you rely on wind, but in the greenhouses, there will be air circulation based on where your flaps are or if you've got fans, for instance. So just making sure that the trap is placed in such a way that the air is always moving through the trap. Okay, this is especially true in the evenings because that's when the moths fly. They fly more in the evenings. Next, I just wanted to have a look at some scouting for FCM in roses. So over here, we've got the eggs. This is what the eggs look like. It's much easier to see on a darker background than on a white rose, for instance. But the eggs do turn color over time. So initially when they're laid, they're quite creamy. And then the eggs will start turning like an orange pinky color as the larvae develops. And then finally, the egg will have a black spot on it as that head capsule is almost finished developing before the egg hatches. So you can tell the age of the eggs based on that. And then obviously, once they start turning that pinky orange color, it will be easier to see on the white or pale roses. Once the little larvae hatch, they'll start to bore into the rose, and you can see here, this is what it looks like when it's entered the rose. So that's the scarring from the larvae that's penetrated through the rose. And you can see here some early feeding on that rose. So just some jagged edges going into the center of the rose. Eventually, as the larvae gets bigger, you'll see it will start to push out frass. So frass is like the excrement from the larvae, and it looks like this sort of brown, clumpy, kind of sawdusty texture. And then this is what it looks like in the stem. So you can see there's an entrance hole here, and then the stem is being fed upon. So don't only check your buds and your roses, but also just double check your stems if you can see that the population is getting high in the orchard. And then finally, this is what the pupae look like. Typically, the pupae will exit and go down into the soil, but you want to try and prevent them from completing that pupation, because if they complete the pupation, then they're going to lead to more generations continuing in the greenhouse. So this is kind of what the systems approach looks like in the rose production. So I showed you the citrus, how the citrus approach looks, and this is sort of translated to roses. So this is that link that I was talking about, making sure that you achieve every step in the link to get good management. And there you can see, starting out with monitoring and sanitation, and then ending off in shipping with the achievements of pre-harvest control measures, greenhouse flower culling, inspections, grading bunches, and then doing final inspections before shipping. So all of these aspects are really important. And you can see here, this is how you reduce the moths. So you start off with a lot of moths, and if you do everything correctly, you end up with very few infestations going out to your clients. So something that's important as we're thinking about sanitation is that one of the best things that you can do to reduce populations is to remove infested materials from your greenhouse or from your fields. So that means cutting or culling any roses that are infested, and then taking them out, mulching them or shredding them or placing them in black plastic bags where they can be solarized. What you don't want is just to leave things out in the open on bare soil where the larvae can complete development and then pupate in the soil, leading to more moths in the future. So really, sanitation is so important. And then also having very good quality control in your inspections and in your pack houses. So making sure that everybody knows how to identify these signs of FCM and that they're removing those from your consignments before they go out for export. Once they remove them, it's then going back again to make sure that those removed plant materials are properly disposed of, as I said, mulching, shredding, or placing in plastic bags for solarization. And I think that that's all for the rose system. I'm sure that there might be some questions that we can talk about later. So next, I'm going to just talk about these integrated pest management tools for FCM. So I've got about, I don't know, 20 to 30 minutes left to go through this. And I'm going to go through it in a little bit of detail, because when we're doing management tools for FCM, it's really important that we're doing it correctly. And there's a lot of different aspects that can affect how tools are implemented in your greenhouses. So the first tool I'm going to talk about is mating disruption. So mating disruption is the use of specific pheromones to attract FCM males, and then using them cleverly within the environment to make sure that the males can't find females. Mating disruption is very highly specific, and it doesn't produce any residues. It's also non-toxic typically. And this means that there's no danger for you to use it in your roses and have this worry that there's going to be chemical residues on your plants. This type of tool really focuses on managing FCM adult populations, And it's an area-wide tool. That means that it acts on the population. So it's not a corrective. It's not something that you can do, you know, in very, very high infestation scenarios to think that you're going to get an immediate reduction in infestation. This type of area-wide technique should be used preventatively. So making sure that you're using it to suppress the population over time. And it really is effective if done correctly. So typically, FCM mating disruption dispensers contain that pheromone, and this is the breakdown of the pheromone. It's different types of dodecynyl acetate, which is the sex pheromone that FCM uses. So there's a lot of different mating disruption products out on the market, and they come in all different shapes and sizes. Some dispensers are twisters like this. Some dispensers you hook onto the tree, then you get your flowable applied ones, and some come in packets, etc. But the way that they work is all quite similar. You'll have your dispenser. The dispenser will release a controlled amount of the pheromone over time, and then through that you get plumes of the pheromone that will help to control the FCM. So in terms of how it works is typically in a normal wild situation, you have a female moth and you have a male moth. The female moth is the moth that emits the sex pheromone, and when she emits the pheromone, it comes out in a plume like so, like this grayish area over here. Now, what happens is the male is much more active in the environment, and he'll fly around trying to detect this plume. When he does detect the plume as he's flying, what he does is he makes a zigzag pattern along the gradient of the plume. So what this means is that he zigzags along that plume looking for the higher concentration. So that higher concentration tells him where the female is so that he can find her and mate with her. Now, with mating disruption, what happens is you've got all of these different dispensers in the environment. And these dispensers are kind of acting like a female. And each dispenser is going to emit its own plume. When this happens, the plumes start to interact, they overlap. So you can see here that the plume is overlapping with the natural female pheromone. This plume is overlapping over here. These two plumes are overlapping. And now, as the male is flying along, he comes into contact with one of these pheromones in the plume, and he will do his normal behavior. He'll start trying to zigzag and find where the highest concentration is. But now, instead of just being able to find one plume, he's finding multiple plumes, and it confuses the male moth so that he can't find where the highest concentration is. He keeps interacting with different concentrations from different plumes, and he's unable to find the female and mate with her. So that's basically how mating disruption works. Now, you do get other types of pheromone-based control. One of these types of control is attract and kill. So there's a little bit of differences between mating disruption and attract and kill, even though they're both using pheromones to help suppress the pest. So for mating disruption, you have overflooding of pheromone. So the goal with mating disruption is to apply it in such a way that the pheromone plumes interact and overlap with each other. For attract and kill, the goal is for the male moth to be able to find the area where the pheromone is coming from. This is because attract and kill products typically have an insecticide in them. So the male moth gets attracted to that point source or to that pheromone, comes into contact with it, gets insecticide on him, and then he dies. And that reduction in the male population prevents mating and reduces the population. So for attract and kill, you spread out your plumes or your dispensers much more sparse in the environment. So instead of having overlapping pheromone, you have this separated pheromone. So there's a couple of ways that you can use this. Typically, attract and kill you want to use on an area-wide method. Mating disruption you want to use on an area-wide method. And attract and kill you can also do an area-wide method, but some people like to use it as an edge treatment. So treating the edges of their orchards and protecting their crops from getting males coming into that area to mate. Okay, now there's a couple of different key considerations when applying FCM mating disruption. One of these is the longevity. So the different types of dispensers that you use, or the different products that are mating disruption products, have different longevity. And that's because different products have different quantities of pheromone. Now, your pheromone load decreases over time, and it dispenses more quickly in warm temperatures. So if you're using mating disruption, I encourage you to read the labels carefully. Make sure that you know how long those dispensers are going to last for, and making sure that you reapply the mating disruption product when you need to. So for instance, our mating disruption product typically lasts about 12 weeks. You have some that are lasting a bit longer, and you have some that are lasting a little bit less. So just make sure that you are applying them when you need to, and that you understand how quickly the dispenser runs out. Something else that you want to consider is where you apply the dispensers or the mating disruption product. So pheromone is heavier than air, and that means that the pheromone sinks. So if you apply the mating disruption over here, that means the pheromone is going to sink straight to the ground, and it's not going to be able to be picked up by the male moths that are flying within the canopy. So for citrus or large orchards, what we recommend is about 1.8 meters to 2 meters. For the roses, what you want to do is hang it about 1.5 meters to 1.8 meters. I do know that sometimes people apply it to the top of their greenhouses, but I would caution against that a little bit. What you really want is that pheromone to be in the canopy where the moths are flying. So try not to hang it too high, because you might not be, with wind movement, etc., you might not be getting the pheromone into the canopies of the rose bushes. So just be careful about how high you hang these things. Another key consideration is weather and application point. So you just want to choose an application point that's going to be firm, where the product is not going to fall off. So whether it is something that you hang, or whether it is a flowable mixture like the splat over here, just make sure that it's secure and that it's not going to fall off, because if it falls off, that pheromone is not going to dispense. Fortunately for most of the greenhouses, weather's not too much of an issue. What I would keep in mind is the temperatures. If the temperatures are getting very hot, just making sure that you're monitoring your traps to know if the product is working correctly, so that you can reapply if necessary. So this is the application that I've seen in roses. So what we're recommending is for our splat product to place it into these dispensers over here, these M3 traps, and then hang them out in the roses. And then obviously you get your other dispensers that you can hang out. So this is just the concern that I have. See how high these mating disruption products are being placed above the canopy. This means that the pheromone will be sinking and then potentially being pulled out by air currents out of the greenhouse, which means that you may not be getting the pheromone here in the canopy where you want it. So something that maybe having a look at as you're doing this is trying out some options for hanging mating disruption on the poles, and then also assessing the differences. So if you've got this scenario versus another scenario, try and use your monitoring to see how well is it actually working. Another key consideration is the effect of slope. So if your pheromone sinks, obviously it will accumulate at the bottom of the slope, right? So if your fields or your orchards are sloped, just make sure that you're placing more of the pheromone towards the top of the slope so that it can sink down to the bottom. Finally, we have to consider edge effect. So as I mentioned, mating disruption is an area-wide technique, and that means that it needs to be applied on a large area. The larger the area you apply it to, the more effective the mating disruption is. And the reason being is that there is an edge effect. Females that mate outside of the area where mating disruption is used can still move into your orchards and then lay eggs. So you want to reduce this edge effect as far as possible, and the larger the area you apply to, the lower your edge effect. So typically, we recommend that if you've got greenhouses, make sure that you apply the mating disruption throughout the greenhouse at the correct rate. So make sure that you're doing it, if it's 500 application points or 100 application points on the label, make sure you're applying it at that rate. And then if you've got any extra stock, you can round them up on the edges to reduce that edge effect as well. Okay, finally, for mating disruption, I wanted to just talk about the role of trap shutdown. So what trap shutdown means is that your traps will catch 0 moths if you use mating disruption. So if the mating disruption is applied correctly, it's applied at the correct time, and you're using the product efficiently, your traps that are placed in your greenhouses or in your fields should catch no moths, because the male moths are unable to find the pheromone trap because they're being confused by all the pheromones from the mating disruption. So a good way to tell if your mating disruption is working effectively is that you should catch no moths. If you've applied mating disruption and you're still catching quite a few moths, either your mating disruption hasn't been applied correctly, or there's an issue with the product. So this is something that you can do as growers, as farmers, to make sure that the products are working effectively. If you have applied mating disruption and you're still catching moths, then I would reach out to an industry expert to try and figure out why is the trap shutdown not occurring. However, trap shutdown, while it can tell us mating disruption is working properly, it can be a bit of a problem because it won't be able to tell us still if female moths are moving into the area to lay eggs. So it's not telling you what the population is actually doing and whether you're still going to get infestation. So we want to make sure that we're monitoring the traps inside our orchards to make sure we're getting traps shut down, but then also potentially using outside traps. And what we mean by outside traps is placing traps outside of your treated area, maybe in some natural bush, maybe in some other nearby orchards, to try and see what is the population doing so that you can still time your foliar applications correctly. So this is something that River Bioscience does in some of the areas where we apply mating disruption. And this is just an example over here. So all of these little blocks that you can see in colors represent the orchards that have mating disruption. And then these circles represent our outside traps. So you can see the outside traps are placed outside of the orchards. And it's just used to understand what is the FCM population doing in the area so that farmers can still time their management practices accordingly. As I mentioned earlier, for roses, you want to do about 1 trap per every 2 hectares for your greenhouses and your fields. But if you are using mating disruption, maybe considering placing some outside traps as well. Next, I'm going to go into foliar products so that I can just talk to you a little bit about types of management that are beyond area-wide management. So these are more spot treatments and corrective treatments. So there are quite a few different foliar options out there. I'm focusing a little bit more on the more environmentally friendly options over here, but you get various different products containing granular virus, which I'll talk about shortly. You also get methoxyfenicide, emamectin benzoate, chlorantraniliprole. You get some other biological control options such as Bovaria bassiana, Metarhizium anisopliae, and then some nematodes such as the Heterorhabditis. So these ones, methoxy, emamectin, and chlorantraniliprole, are synthetic chemicals. These two are fungi. As I mentioned already, this is a nematode, and then the viruses fall under the virus class. Now, as a member of River Bioscience, I'm not as familiar working with these types of products. So I'm not going to talk about them in too much detail. I'm going to focus more on the virus products because that's my area of expertise. If you are looking to use some of these other synthetic products, I implore you to reach out, get the labels, make sure that they're correctly registered for your area, and then use them accordingly. Some of the information that I'm going to talk about with regards to granular viruses will be relevant for how you apply some of these other products. But as I mentioned again, use that product correctly and make sure that you're stewarding it, because if you don't do it correctly and just follow those labels, you're actually not going to get that efficacy that you're looking for to reduce your FCM. So as I mentioned, I'm going to focus more on virus products. So the granular viruses for false codling moth contain the Cryptophlebia leukotrieta granular virus. So this virus, as a foliar application, is very highly specific. it only targets FCM. This means that if you've got other moths in the environment that are problematic for you, then I wouldn't recommend a virus application. What I would recommend rather is to go for one of these more broader chemical based applications if you've got other moths. I don't want you to be applying virus thinking that it will control all of the problems. This virus will control false codling moths specifically. So, just making sure that you are doing your monitoring and that you're identifying your pests correctly. So the nice thing about viruses is that they manage the FCM larvae, the little neonate larvae, before they actually penetrate the crop. So it can help to protect the crop and prevent damage. So how do viruses work? Well, typically you'll spray the virus. You can use knapsack sprayers or mist sprayers, and you'll spray it as a full cover spray on the crop. It's very, very important that it's used as a full cover spray. And this is because the virus is only effective if it's ingested. Some of the other products that you will use, such as the fungi or synthetic products, also need to be sprayed as a full cover spray. So anything that's targeting the larvae as a contact spray or an ingestion spray should be full cover. If it's systemic or if it's killing the larvae in a different method, then make sure you're just following those label rules on how to spray it. So the virus is sprayed onto the crop and then it covers the surface of the crop. Then the tortoisid larvae, the little FCM larvae, will hatch out and they'll begin to feed on the surface of that crop. So either on your flower buds, on the leaves, or on the stem. And when they do this, they will ingest the virus. So the virus will go into their stomach. Once the virus is in the stomach, it signals to the virus that it must unzip. So viruses have a little protective protein layer on them. And when it enters the insect guts, the protein layer kind of unzips, it dissolves, it releases the virus into the insect's gut. And something that causes this protective layer to dissolve is the fact that the insect gut is very highly alkaline, so it's got a high pH. Once the virus is released, it gets absorbed into the little microvilli inside the insect gut. So these are little cells that allow for absorption. And then they enter the cells of the insect and they start to infect the insect. So the virus enters a cell and then that cell replicates the virus. So the virus multiplies and multiplies and multiplies. It infects more cells throughout the insect, and then eventually the larvae will die. So there is some, basic things that we need to consider when we're applying viruses. So you want to apply viruses immediately after you mix it in your spray tank. You don't want it to be sitting in the spray tank overnight because there is the possibility that little protein layer that protects the virus will start to degrade. As I mentioned, you really want to apply as a full cover spray with mist blowers or handheld sprays. And ideally, you want to apply in the later afternoon, evening, or early morning to reduce the UV exposure of the virus. This is because when the virus is sprayed, it's suspended in the water droplets, and water droplets can refract the light so that the virus is exposed to more UV during the day. So when you spray it, you just want to give the virus time to dry on the roses before the heavy UV of the day, so midday sun exposure. Luckily for greenhouses, there is a little bit more protection from UV, but I would still be cautious about spraying midday. It really does work best if sprayed late afternoon, evening, or morning. Then you want to make sure that the pH of the water is between 5 and 8. So a lot of different insecticides that you will use as foliar applications have recommendations for what your pH balance should be. So that's also something to check on labels. But for viruses, between 5 and 8 is important. If you go above 8, it's starting to be very similar to the insect gut, and then that virus will be released. If it's below 5, that's a bit acidic and it can start degrading the protein layer. So it's not going to make the virus totally ineffective, but instead of getting 80 to 90% control of your infestation and your neonate larvae, you'll be getting, 70, 60, 50% management. So that's why pH balance is important. And to achieve good pH, you can apply with a buffer, And then you can also apply with a wetter to make sure that it's spreading and getting you that full cover spray. Now, how you apply viruses is very important. And actually, this goes for a lot of foliar applications because you want to try and apply it corresponding with the moth flight peaks. And this is why monitoring traps are important, and especially monitoring traps that are outside the orchard if you're using mating disruption. Because if you don't know when your moth flight peaks are, then you can't apply it effectively. Otherwise, you can use it in a calendar program. So that means just reapplying the chemical or the virus, repetitively throughout the season. And I think with roses, that would be every 1 to 2 weeks, depending on how quick the roses are being harvested. So in terms of virus efficacy, there has been a couple of questions about this because I think some people did some testing with virus in roses quite a while back, and they said that they didn't get good control and therefore weren't going to use viruses. However, if used correctly, viruses have the same level of efficacy as any other of the registered products, including synthetic chemical products. And the viruses also have good length of durability in the environment, giving you control for between 4 to 12 weeks. So viruses are as effective as other chemical control products, but you need to use them correctly, and that's why I was just giving you this summary over here. And especially the full cover spray, because if you're not reaching that target area where the larvae are hatching and ingesting and penetrating the crop, then you're not going to get good efficacy. Another key consideration is to make sure that you are reapplying when necessary. So like any other chemical, viruses do degrade over time. And as you get closer to that 4 weeks, you start to get lower levels of efficacy. So depending, like I said, on the timeline of the roses, you want to make sure that you're keeping the virus level high within that crop and targeting those larvae on your buds and on your stems. So whether that means a 1 week to 2 week reapplication or a 3 to 4 week application, you're trying to protect those crops that you're going to be harvesting. So as I mentioned, active monitoring is important, and you want to generate trap catch data according to False Codling Moth monitoring standards. Typically, if you're going to apply according to flight peaks, you want to apply chemical products and viruses about 7 to 10 days after flight peaks. This is because once the moths have peaked, they begin their egg laying, and then you still got a little bit of a delay before those eggs start hatching. So you want to time the virus sprays approximately 7 to 10 days after that peak, and then you'll catch that early hatching and prevent the larvae numbers from getting high. So this is something interesting I really wanted to show you, and it's how do we use virus together with mating disruption or area-wide management to get good control. So this doesn't only have to be virus, it can be other foliar applications as well. But really, it's just showing you why it's important to use different types of management methods. So this line up top here is demonstrating an area-wide pest management tool that is applied at the beginning of the season in citrus, and then is getting reduction in FCM pressure. So if you use area-wide management by itself, you will get a reduction in pressure, but it will take a little bit longer to achieve good pressure. And throughout the season, you won't get as good reduction in infestation as you would if you used it with really well-timed foliar applications. So what we call this is a knockdown effect. So when you use a foliar application in conjunction with mating disruption or another area-wide pest management tool, you get a knockdown effect. Your foliar application reduces the FCM moths and numbers quite quickly, and then it gives you more opportunity to apply your mating disruption or your area-wide management in a way that allows it to work more quickly, because the lower number of the population that you get initially when you apply the area-wide management technique, the faster that technique is going to work. So I hope that that makes sense to everybody. But basically what you're looking for is well-timed foliar applications together with area-wide pest management to help you reduce the population very quickly to get to almost a zero threshold. Okay, to round up my discussion, I'm just going into biological control as a third integrated pest management technique for false codling moth. So we've already talked about area-wide management with the mating disruption and attract and kill. Then we spoke about foliar applications as correctives, and now we're going to talk about biological control. So biocontrol, as I'm sure many of you are aware, is the use of a living natural enemy to suppress or regulate populations of a perceived pest through predation, parasitism, or disease. So this is microbial biocontrol in particular that I'm going to be talking about, because viruses and fungi, for instances, are other types of microbial biocontrol. So biocontrol in general is any activity of one species that reduces the adverse effect of another species. So for microbial control, what I'm going to talk about today is augmentative biocontrol. So you get other types of biological control for microbial control, and these include classical biocontrol or neoclassical biocontrol. But augmentative biocontrol is very different in relation to these other ones, because this involves the mass rearing and release of biocontrol agents to control pest populations when natural enemies in your cropping system are insufficient in number or aren't able to get good control because the crop system is not suitable for those biocontrol agents to get to high numbers in order to reduce the pest population. So the goal is in augmentative biocontrol to apply greater numbers of that agent than what is naturally present. So you get inoculative control and inundative control. So inoculative control is when you do multiple smaller numbers of releases of enemies over an extended period of time to try and get their populations consistent and higher. And then inundative biocontrol is when you release a very large amount of natural enemies to quickly reduce the pest population, and then you can come in and do your other management techniques. So the biocontrol agent that I'm going to talk about today is Trichogramma toidea cryptophlebia. So this is an egg parasitoid of tortricid moths. So this one not only affects false codling moth, but it can actually impact other tortricid species that may be in the environment. So with this egg parasitoid, what they do is that the adult little wasp over here, these are the adults, there's a female and a male. The female has very smooth antennae and the male has kind of hairy antennae, and that's how he picks up the female. He can detect her pheromones. So what happens with these adults is that they mate, and then the female will go and lay her egg within the egg of the pest species. When her egg hatches, that little wasp larvae will start to feed on the contents of the pest egg, and then it will kill that target pest and prevent it from hatching. So you can see this in this life cycle over here. You have this little wasp. She lays her egg inside the FCM egg. Then the little wasp larvae will hatch. It'll consume the entire contents of that FCM egg. She will then, or the little larvae will then pupate, and the egg will turn black. And this tells you that the FCM egg has been successfully parasitized. After a couple of days, the adult will then emerge, and then the life cycle will repeat itself once the adults have mated. So these parasitoids are typically active at temperatures between 13 to 35 degrees Celsius. I know the greenhouses get a lot hotter than that, but it doesn't mean that the parasitoids will die. Instead, the little wasps will find areas within your crop to sort of hang out for the day while the temperatures are too hot for them, and then they will emerge later on when the temperatures cool down to go and parasitize eggs and mate. And then every female can lay up to 70 eggs per FCM population. So for the application of these biocontrol agents, what you want to do is apply 20 to 25 cards per hectare, with each card having approximately, or on average, 1,000 to 1,200 parasitized eggs. And then the wasps will emerge from these eggs and go off into your crops to find new eggs to parasitize. So for applications, what you want to do is apply the cards evenly. You can take into account your tree spacing or your rows spacing, or maybe your beds, for instance, to determine where the cards need to be placed. And the important thing is just to make sure that it's even. The little cards that you get for these Trichogrammatodea should be handled a little bit delicately. You don't want to press them too hard because it will squash those eggs and it will kill the little wasps inside there. And then you want to place the eggs sort of on the bottom side of the leaf inside the canopy, and then just not too dense though. So not right in the middle of all the leaves, but somewhere on the outer edge on the lower side of the leaf. And then you would staple them onto that leaf, or there's like a little sticky section that you can wrap it around the stem of the leaf as well. And then try to place in the morning or afternoon so that the wasps have time to acclimatize and then they're not exposed to heat shock. So something that I didn't mention about this wasp is that it is a native species. So it's a native pest parasitoid of FCM. So you do find natural populations of this wasp. And with these inoculative releases, what you want to do is just increase those populations so that you get better management of the pest. So this is what the eggs of the FCM look like. So this is how they look when they're first laid. They're kind of creamy. And then what we do is we expose these FCMs to parasitoids, and then the parasitoids parasitize those eggs. And then the little parasitoid larvae start developing in the eggs, and the eggs turn a reddish color. If you are looking in your fields for eggs that have been parasitized, this stage can look a little bit like the FCM egg is developing in there, so that the FCM larvae is developing. But for parasitized eggs, this egg is completely red. Instead of seeing that sort of C shape that you sometimes get with FCM developing in an egg. But eventually, these little parasitoids will start to pupate and those eggs will turn black. So if you're looking in your orchards and scouting for parasitized eggs, you can see these very easily, and then you know that the parasitoids are in your greenhouses or in your fields. Eventually, the pupae will mature, and then the little parasitoids will start to emerge. So when they emerge, they chew a little hole in the egg, and then the wasp comes out. And this is what the egg sheets look like once all of those wasps have emerged. And you can actually see this in the field. So if you are scouting for parasitized eggs, you can actually see when the wasps have emerged because you can see those little holes, especially if it's on a lighter background like a cream-colored rose. In order to get good efficacy with wasps, you want to do multiple applications because this will help you to get the population higher and eventually give you better control with over, in this instance, over 51% of the eggs being parasitized over and above natural levels of parasitism. And then from this graph, you can see as parasitism increases, so does infestation. And now if you're using it in an integrated program, you'll get even further reductions with your foliar sprays and then hopefully your area-wide management as well. Something to consider when you're applying biocontrol agents, such as Trichogrammatodea, is that other types of chemicals can have an impact on the populations of the Trichogrammatodea and the success of your biocontrol agents. So anybody who wants information on this, I've got a whole list of different types of chemicals and their impacts to biological control agents so that you can sort of understand how to time applications of biocontrol agents to get best efficacy. But at the end of the day, what we want to do is mimic what we've done here in South Africa for our FCM control. And we want to do this in roses in a holistic way. So trying to use multiple different types of tools for integrated pest management at various stages of the life cycle of the pest in roses to get adequate pest population suppression, reduce the infestation in the roses, and allow for better export. So with that, I would like to say thank you. It concludes my talk. And I do see that there have been a couple of questions coming through, which I think we will likely address after Mary's discussion. But yeah, it's been great to talk to you, and it really is an honor and a privilege to be able to learn a little bit on how FCM affects roses and to help you achieve better pest management using a systems approach. Thanks, John.
Good. Thank you so much, Megan. Quite an insightful presentation.
No problem.
I think talking about FCM, South Africa have the most experience all over the world dealing with this pest, particularly in the export market. Thank you so much. So once again, for those just joining or joined late, welcome to our webinar session. At this point, I'd like to welcome our second speaker, and that is Dr. Mary Guantai, who is going to talk to us about the regulator's perspective to FCM. Where are we and where are we likely to go? Is the situation so bleak, or is there light at the end of the tunnel? So Mary is going to talk to us about that. And once again, just to clarify a few of the ground rules, the presentation will be available to you. I can see questions coming up concerning that. Will be sent to you after this. And also use the chat box to ask as many questions as you can, but there'll be a Q&A also at the end of it all. Karibu, Dr. Mary.
Thank you so much, John, for that. And thank you, Megan, for the presentation. It was very good, very impressive. We have learned a lot.
Thank you, Mary.
Yeah, so, I hope you can see my slide. I will start my presentation. My name is Mary Guantai. I work with KEFIS. KEFIS is the National Plant Protection Organization for Kenya. And we are a regulator. Our work is mainly to plant health. We undertake plant health issues, including taking care of phytosanitary services. We assure that plants, pests, and diseases are not able to get into our country or get to other destination where we have our markets. And we also undertake seed certification for our inputs, the seeds, and we assure also that they are safe for use by undertaking analytical services at our labs. And also we protect the breeders' rights by having the plant protection role that we undertake at our headquarters. And so in general, we are focused on plant health, and we work through various regulatory frameworks. One, we do our official controls guided by the KEFIS Act, number 54 of 2012. We also have our Plant Protection Act of 1972. We have the Seed and Plant Variety Act that guides us in a seed certification process. Plant Protection Act, basically most of it has a lot on phytosanitary aspects. The Agricultural Produce Act. We also have legal notices which guide our charges and other things that we need to implement in the industry. We have international laws and regulation that guide us, including the International Plant Protection Convention and also the international standards for phytosanitary measures that guide our work. So Megan has spoken a lot on the False Codling Moth. My presentation is quite short, so I do not want to deliver on this. But just to mention that in Kenya, the False Codling Moth has been a major pest of concern to the industry, particularly on the roses and people who are doing capsicum, because we've seen that most of the interception that we are getting from abroad or from the destination market has been mainly on the roses and the capsicum. So just to take you through the status of the rose cut flower since when we began this challenge. Since 2018, this is where we realized that we have a problem in the roses. And this came because we started getting regular interception of the rose cut flower due to FCM. And as a result of that, in 2019, the EU implemented the regulation 2019-2072. And this regulation, though it didn't have any additional declaration for rose cut flower, but the FCM was regulated, and therefore there was zero tolerance for FCM to the market. Therefore, we had to make sure that consignment leaving the country were free from the pest.
Sorry, Doc, can I interrupt you?
Yes,
the
please.
slides don't seem to be moving.
Really? You cannot see
Yeah,
it?
I think we are stuck on the first one still.
Oh, sorry. Maybe I
Yeah,
can stop
yeah, it's
sharing.
now moving.
Then
Oh, it's okay.
sharing
No, it's
again.
okay. No, it's okay.
Oh, it was okay. Sorry.
It was okay. You can move from the left. I think it's not showing the presentation mode.
It's not showing the presentation mode.
Yeah, it's not showing on the presentation mode. So
On
we are
my
now
slide?
seeing number 4, rose cut flower FCM status.
Yes, right. That's where we are.
All
Yeah.
right.
Sorry if
Please
you cannot
carry on.
see the presentation mode. I don't know what happened, but from my side, it is showing the presentation mode. Yeah. So I was saying that once in 2019, the regulation came into force. Then in 2020, there was a follow-up by the EU regulator. The DG Sante came in, and they undertook an audit in our farms on the cut flower roses to check what was happening, what kind of measures were we implementing. And the conclusion of their study, or the conclusion of their audit, was that the rose cut flower was still posing a risk to introduction to the EU. And therefore, we needed to have more stringent measures to ensure that it doesn't get into consignment. So come 2023, there was another follow-up whereby the EU wanted to know how far we have gone in eradication, quote unquote, but they undertook a risk assessment to check how we are faring, because they realized that still we had high interceptions and from Kenya due to FCM. And the European Food Safety Authority, that's the EFSA, came to a conclusion that the introduction of FCM to the European territory was still possible, because the FCM was still available or is still present in our farms. So in 2024, as most of you know, we have seen additional sampling. Initially, we had from 2018 to 2020, we had 5% sampling. Then from 2018 to 2019, there's 5%. And from 2019 to about 2023, that is in April, we had 10%. And now currently the sampling is at 25%, which means that there's higher risk because there's higher possibility of detection of the pest in the market. So now, yeah. So now we check on the trends of interceptions, and you realize that, I hope you can see it from the from below, where we have the years from 2018.
Can I share from my end? Doc, it's not showing. Can I share from my end?
Please
Yeah,
share.
let me do. Let me do.
Stop
Please.
sharing. Yeah.
Yes. It's clear. Sorry, let me just get this right. Just 1 minute. I'm sorting it out, Doc.
No problem.
Please let me know if you can see it from your end.
Yeah, I can see it now.
Oh, that's the good.
Yeah.
Good.
Yeah, so that's where we are. I cannot see the years from my side. Yeah, I think they're not very clear. Okay, thank you. So from 2018, as you can see, we had quite high number of interceptions due to FCM. By the end of the year, we ended with 48. 2019, we had a further rise after the stringent measures were introduced. And again, we had some slight drop in 2020. Come 2021, more stringent measures were applied in the industry, by the exporters, and we realized that the population went down and further down in 2022, where we stopped at 23 interception. Last year, we had a rise in the number of interception, where we had 28. 21 due to from roses, and on 7 were from the capsicum. And now currently we actually standing at 40. 38 is due to roses alone, and so we have 2 for capsicum, and therefore you can see that the trend is that the interception are increasing. Next, please. If you compare these years that in terms of interceptions and the rejection that we have, you can see that we are really doing quite a lot to prevent this pest from getting to the EU territory through the rejections that are implemented at the exit point. For rejection, what it means, the difference between the interception and the rejections is that the interception is what is rejected abroad, and rejections are what we intercept at the exit point. And so we have more rejections at exit point compared to the interception. And you can see, like this year, we are having like 72 rejections that have been registered compared to the 38 interceptions for roses that we have currently. Next, if we compare the false codling moth with other pests of concern to the market, we still realize that the false codling moth is still giving us high interception. It is still the most problematic pest to the market since 2020 to date. And so we have others like thrips, which are coming in, especially from France. We have a number of interception from France due to thrips. We have whitefly, we have the fall armyworm, the leaf miners, and a few others. So basically, the slide is to show us that the false codling moth is the major challenge to us, and we need to make sure that we control it. Next, please. So in terms of the market, we find that the Netherlands is the major challenge, or is a market that where we are getting more interception. And this is where the EU has clearly said that they do not want the FCM into their territory, followed by France. France, we have seen a number of interception due to FCM and Highland. We have a few other countries, but this one has been a bit manageable, so we cannot compare with what we are getting from the other countries. Next, please. So in terms of the produce that we exported, we realized that roses has been the major challenge, and it has been the most affected by FCM, followed by osimum, the basil, and some plant for planting. And also capsicum comes number 4. I mentioned that capsicum also is a major problem, and currently we are having the pest-free places for production to ensure that the pest does not get in. Next. So we have a number of interventions that are undertaken by the industry together with KEFIS. And one of them is the meetings that we've had. We have had a number of engagements with the stakeholders. The stakeholders include the Kenya Flower Council. We have the Union Flyers, which have helped us to engage with the industry on actions that we can take to make sure that the non-compliances are reduced. We have development partners like COLID who have come on board to even sponsor some intervention measures, including capacity building for the staff in the for exporters and also for inspectors. And we realize that the growers are now much more committed to ensuring that the pest is contained, including self-regulation and also having joint fora, which helps the agronomists or the people who are present to be able to share information on how to manage FCM further. We have seen a lot of investment in the industry, and this has to do with ensuring that we have plastics. We have the covers that are there installed. We have the nets. People have put the net so that when they roll up the plastic, then we protect the pest from getting in. So there's been a lot of investment on the part of the industry. We also next we have done registration of exporters, and this is to make sure that we know who we are working with, and whenever we have an non-compliance registered in the market, we can quickly trace to where the non-compliance emanated from. And so before we register the exporters, we make sure that we undertake a farm system audit, and before we are able to access the account. So we train the exporters also, because we make sure that when we register the exporters, they are able to use our electronic certification system, and so we have to train them on how to do the submission through the system, so that we can also issue the phytosanitary certificate, and thereafter we have our phyto be sent through the e-phyto. Next, please. So we will undertake field farm inspection and also system audits at the various places of production. And this is to make sure that the pest control measures that the industry is implementing is in place, and we are able to contain the pests from right from production, as Megan was insisting on the systems approach, right from production to the exit, so that we minimize the chances that this FCM can be found in the consignment. And so we undertake this scheduled farm audit, and we make sure that all the basic tools that are required are implemented. For example, if it's monitoring tools, we make sure that they're doing it properly. Next, please. Next, this is just an example of an inspection that is being undertaken. So at the exit points, this is where we have to undertake some various measures. We had to increase the sampling and the inspection at the exit point, since we realized that the pest is becoming problematic. We have tried to do some risk profiling for companies, such that we categorize them based on the number of non-compliances they have got. So we have those that we regard as low risk, medium, and high risk, and this has enabled us to do sampling higher than what is being done on the other side of the importing country. And we are also guided by the International Sanitary and Phytosanitary Measures number 31, which helps us to undertake good sampling by making sure that we sample based on the various strata that we have. That means we have a lot. If it's a consignment, then we can be able to separate it into various strata to make sure that we obtain a sample that is appropriate from each of the strata. And therefore, if we get the non-compliance at this point, then there's a rejection that is undertaken, and the farm is allowed to undertake corrective action before they can resume export. We have undertaken capacity building for both the inspectors and also the staff at the production points, whereby we have created a lot of awareness in terms of detection and identification of the pest and also management. The industry has also taken a key role in this, and also they have tried to understand the market requirements through the various forums that we have held with them, and also in-house they have tried to also make sure that this has been passed through. We have various protocols that have been developed. One of them is the systems approach protocol that is guiding us on FCM management, and a number of other protocols that we are using to ensure that we manage this pest. It's just training and capacity building of the inspectors and also for the industry. Just a few pictures showing that. Yeah, so I said we are using the electronic certification system, and this one is the system that we use to enable us to issue the phytosanitary certificates. And so our system is connected to the EU traces, whereby we are able to exchange the phytos through the hub. And after that, the phytosanitary certificate can be transferred electronically to traces. Therefore, as we send the hard copy to EU, we are also able to send the e-phyto. And this one protects or make sure that we take care of some of the challenges that we had, including faking of the certificate and so on. Next. We have enhanced or trying to implement the greenhouse infrastructure that is good enough to make sure that the pest does not get in from outside. And therefore, the industry and the exporters have done a great job in making sure that they cover the greenhouse well. We have greenhouses which are well covered with plastic, and also, as I said, they have side nets. Some have gone to an extent of even covering the top vent to make sure that the pest does not get in through. We have double doors which have been installed such that somebody gets into the greenhouse, you open the first door, you get in, close the door, then are able to get into the second door to make sure that just by opening the door, you are not leaving it open so that the pest does not find its way in. We have mechanism for humidity management which have been in place because one of the things that makes most of the people roll up their greenhouses is because of the humidity. And so people have trying to implement other measures to make sure that the humidity is taken care of so that we do not have the temptation to open the side vents. And therefore, we have fans and other methods of making sure that the humidity is managed. So these are just examples of some of the greenhouses or what we are having as now our production sites, which are good enough to make sure that the pest does not get in. We have a lot of quality control that is being undertaken, particularly at the intake and also within the pack house, so that they have gadgets which enables them to be able to see. You can see we have enough lighting, we have white tables. They even have some pest cards which can enable them to be able to detect the pests that they are seeing or whatever they are seeing, to be able to enable them to understand more. And so they have a lot of investment in terms of the quality control along the chain. We also have farms which have some dispatch. Before dispatch, we have an online QC. We have the quality control before dispatch, and also have a team that that's the pest management team that has been put in place in some farms to make sure that they check everything before it leaves for the airport. So FCM management, this one I don't want to stretch to talk much about it, because I think Megan has talked about integrated pest management. Here we have a lot of regular monitoring, and the monitoring is undertaken through the use of pheromone traps, and also have scouts in all the farms who undertake monitoring to make sure that any count can be taken care of. They have protocols, especially the integrated pest management protocol for FCM, which is in place. And they have mechanisms to make sure that any non-compliance is taken care of quickly. The scouts are able to report quickly so that it can be able to inform whether there's need for spray or any other way of dealing with it. And at the exit point, we have made sure that if we detect any pest at the exit point, then this is treated the same way an interception is treated, to make sure that we become more stringent in the way we handle our pest management. So what we expect from this, one of the things that is coming in next year, 26th April, is that we are having regulation on roses. So coming next year, we have more additional declaration. From 2020, we didn't have any additional declaration for roses, but come next year, the regulation is coming into force, whereby roses will have more additional declaration. We will be required to put in more stringent measures. That is something that is coming. We have intensified pressure for the controls at the exit points. That's why I said we have had to do risk profiling to make sure that we even undertake more sampling compared to what we were doing before. We have we realize that with more pressure that we have, it takes more time for the consignment to leave the exit point. And that means that we have pressure on the logistics, because people have flights that are leaving, and so there's more pressure that is coming in because of that. More costs also, we have handed costs because consignment have to come earlier. We have a lot of measures that have been employed along the supply chain. We have a lot of quality issues, because more sampling means that more boxes are destroyed or more boxes are opened, and this can lead to some quality issues. And we also have a possibility of reduced market share of, or loss of the competitive advantage because of the numerous interceptions that are experienced from our side. Next, please. So we have learned that there's need to have a good way of detecting the pest early enough. And this is very important by making sure that the capacity of those people who are handling the consignment, those people who are handling the produce at the production site, the scouts or the harvesters and all the staff are informed on this. Then the farms are employed an integrated approach in management of the pest to make sure that it is completely, if not completely eradicated, but we have minimized the detection level. We have done a lot of inspections, and we realized that the farm system inspection approach is very effective because it enables that we are having good controls from the start, and that means that towards the end we have less risk. We have observed that the pest works with a kind of season whereby in March and June more of the detection have been noted, and therefore these are the months that we have been more stringent to make sure that we do not have high interceptions. And the facilities have been well maintained, as I've talked about the infrastructure to make not getting. Next. Yeah, so the challenge have had to deal with is the tropic weather conditions, favorable weather for the pests. Sometimes we have found challenges with that we are applying, particularly in cases whereby we have strict regulation from the, and so some categories of the pesticides are not allowed. Difficult to control using the current measures. John, you put me off.
Daktari, please share your
Yes.
presentation, and then I'll take control. I'll move the slides for you.
Okay, I'm just finishing. Actually, I was.
Yeah, you can share and then I'll move the slides for you.
You can see it now. Yeah, it's number 9.
Sorry, I think I had a call and it disconnected. Can you get back, or Lewis, are you able to control it from your end? I can continue
Can you see
sharing
it
from
from your
everywhere.
side?
Sorry, I don't
John,
have
can
the
you see?
option. I don't have the
You
Can you
don't have
see?
that.
Yeah.
Yeah, let me share. It's okay.
Okay.
I'm sharing. I don't know whether you can see it. Can I stop sharing then?
No,
Yeah.
Can you see? That's we're on 9. We're on 10 now.
Yes,
Doc?
John.
Yes. Yeah. So this is actually my last slide. This is on the way forward. So we have tried to make the deliberate effort to make sure that the Kenyan produce is safe and free from FCM, so that you can be able to meet the EU regulation and be able to protect the market. We appreciate the support that we have received so far from EU in terms of capacity building for both the inspectors and also the private sector. And therefore, we invite all the investors in the country on various ways of mitigation of the pests. In Kenya, the major challenge that we have is on eggs, because more than 90% detections that we've had to the EU have been due to eggs. And therefore, it would be great to hear from Megan, for example, what they are doing to make sure that they can be able to see the eggs, because the detection, we have some gadgets which can help in the detection of the eggs, which can be able to be used by the people or on the farms, and also whether we have some chemicals that can work particularly on the eggs, because currently we don't have so many ovicides which can be able to help in control. I think that's where I stop. Thank you very much.
Thank you, Doc, for the wonderful presentation. It gives great insight into what is expected from the market side and from the regulator side. Thank you so much. At this particular juncture, we'd like to welcome the Q&A. Probably we start off with Megan. If you've gone through the Q&A, the chat box, anything that you'd like to prioritize and start off with?
Hi, John. Yes, sorry, there was just some building next door, so I quickly had to tell them to be quiet. So I can see the Q&A chat box, but I'm having difficulty accessing the normal chat. It's not allowing me. So I don't know if there's any questions in the normal chat, but I can address some of the questions in the chat box if that would be preferable. Can you hear me?
Sure. Please go ahead.
Okay. So I don't know. Can you see my video now?
Yes, clearly. Thank you.
Okay. Hi, everybody. So just looking in the Q&A box, I'm just going to go maybe from the first one, I think, which was Johannes. And the question was, if FCM is native to sub-Saharan Africa, where is the pest originally from? So it is native to sub-Saharan Africa. It was originally found or identified in South Africa, but a lot of people suspect that it is native for the region. And a lot of the types of native trees that they feed on, might have been the original host plants that they were able to feed upon. I do have a list that was published on approximately 80 different host plants, which includes ornamentals, it includes, various different crops, and then a couple of native plants as well. So I can share that if you are interested, especially if people are concerned about any hosts that might be around their fields or greenhouses. But yeah, so people suspect that it is native to the entire region.
Okay. Then what I see here, there's a question that says, what is the morphological difference between FCM and FCM? And I'm not quite sure what the person meant specifically on that. Is there another pest that you are wondering what the differences are? Maybe that
Yeah, I saw that, and it's also anonymous, so it's not easy to pinpoint to whom, but it's not clear. Probably we move on to. There's an interesting one, Megan, where I think it's from Mohammed, where he's mentioning that they're using mating disruption within the greenhouse, but the trap catches are still in the delta trap is still high. If you could shed some light to that kindly.
Yes, so I can't actually see that question on my side. But so this is where I get a little bit concerned about how the mating disruption is being applied in the greenhouses. I suspect that if you are hanging them very high, or if you're not placing them at the correct density, that the pheromone is not reaching the target areas of the crop, and then you might still see some of those trap catches. So I think ideally, maybe for Mohammed, maybe if he can give some more input, it doesn't have to be in this forum. They're welcome to email me, but maybe a little bit of input on how they're placing out the mating disruption, and then at what density, which product and how many contact points within that area. Because with mating disruption, you need to get the pheromone to where the moths are flying in order to actually disrupt them. So I hope that it's not really a full answer to the question, because a little bit more information on how they're being used in that specific scenario would be needed. But with some of these points of application being so high in the orchard, and with air movement coming through in the gaps in those greenhouses, it might mean that the pheromone is just being pulled out the greenhouse.
Good, thank you. I think we'd open the door for more engagement with Mohammed at a personal level, just to clarify some of the issues. Then the other one that I pointed out is on the favorable environmental conditions for FCM. I think that is from Mesfin and a few other people in terms of temperature.
Yes, so that is a good question. FCM is a very interesting pest because it tolerates quite a wide range of temperatures, everything from about 2 to 6 degrees up to 40 to 45 degrees. Also, obviously, in the greenhouses and in the cropping environments, there are microclimates where the moths will hang out during the day while they wait for the ideal flying times. But yes, it prefers a very wide range of temperatures. The most ideal conditions are probably about 25 degrees to 30 degrees. At those temperatures, the generation turnover is quite high, and they're happily to be able to fly at those temperatures and mate and lay eggs, etc. The flying, even though it can withstand cooler temperatures between 2 and 6, usually at those temperatures the moths will be quite immobile. They won't be moving around a lot, but they will start to have these large moth peaks and moth mating times anywhere from about 12 degrees to 16 degrees Celsius. So that's usually when you see the moths picking up in population numbers when you're just coming into that sort of early spring here in South Africa. Obviously, I think you guys have a lot more warmer temperatures, so you likely have a moth population that's quite standard throughout the year and very much overlapping.
Good. And while at it, I think I've got some experience with that as well. And this could also be relevant. I think it's also come up on the Q&A, whereby following the placement of the traps, we actually see an upsurge of FCM eggs and damage in the greenhouse. I couldn't quite understand. I thought the male pheromone, the female pheromones should be attracting the males only. But
Yes,
there was that observation that we've noticed. If you got any insight on that as well, kindly, Megan.
Yeah. The pheromones that we use are specific to the males. When you mean the traps, John, are you talking about the monitoring traps, or are you talking about the mating disruption dispensers?
Monitoring traps.
Monitoring traps, yeah, that is unusual. The monitoring traps are only for the males. Could it not be that when people are starting to place the correct monitoring traps in the correct way, that, as we start looking at this as more of a systems approach, people starting to do more scouting and starting to notice things a little bit more intensively? Or is it really an increase in infestation?
Maybe it was coincidental. It doesn't really make sense in this
For
case.
instance, my experience with this, that what I'm talking about is our work with macadamia nut borer in the macadamias here in South Africa. In the past, they would just use very harsh chemicals throughout the season, and they weren't too concerned about that there weren't very much residue limits for the macadamias. Now, as the residue limits are becoming more problematic, macadamia prices are low, the pests are more of an issue, people are starting to monitor, and as they're starting to monitor, they're more present in the environment, and they're looking out for infestation and eggs more in a more committed way. They're just doing monitoring better. So initially it seems like the pest numbers are increasing, but it's not necessarily the pest numbers increasing, the monitoring and the identification of those pests is just better. So maybe, just if you are still concerned about this, we can definitely talk about it. But as we are seeing increase in infestation, just think about, okay, is it because we've got more scouts out here? Is it because we're more on the watch for it? Or is it actually truly an increase in infestation? In which case, it might be the female moths are coming in from other areas. But I don't think that it would be directly related to the monitoring trap themselves.
Thank you, Megan. Then there's another one from Catherine. She's asking, in case of mass trapping, what is the recommended number of traps per hectare? I know this more for the orchards. What would you recommend
For the
experience?
it's quite difficult for FCM because it's not something that we do a lot of. I can maybe, I don't want to give you a number that's false, so maybe what I can do is just have a quick look at that and then share it with the group on the mass trapping. Like I said, it's not something I have too much experience with because in South Africa, we're mostly using the attract and kill or the mating disruption. Yeah, if there's some other questions of regulatory nature in a short while, then I'll look that up in the meantime and then I can share it with the group.
Great. And we can always continue the discussion anyway. Then the other one I think that was interesting is also the distance traveled by the pheromone. I know that's also a sensitive one. And probably while tackling that, there is also the distance traveled by we know it's not such a good flyer, but what the distance traveled by the moth?
Yeah, the moths, they are really not that great dispersers compared to other tortricid species. So I have done a little bit of work on this with our SIT program, and the moths do disperse, anywhere between 60 meters to about 400 meters, just with our trapping networks. I think in open systems where there is a lot more wind to be able to move those moths around, they can actually exceed that, distance. But typically what we see is about a 600, 400 meter dispersal in flight.
Thank you. The other one came from Dr. Guantai, and I think she mentioned it at the tail end of her presentation. The key issue, particularly for the rose farmers, is the egg stage.
Yes.
Is there any way of enhancing identification of the eggs?
Yeah.
And also, what options are there for the management? I know we talked about the parasitoid that we're working to get available in Kenya.
So the eggs, it's quite difficult to not only identify but to scout for them because they're quite cryptic. They're very small, quite flat, and until they start turning color when the eggs are developing, it can be very difficult to see them. For the management of it, there is some information out there that methoxyfenicide can have an ovicidal activity. So perhaps look into that. But it is really, more targeted towards the larvae. And then obviously there is some, additional, there's obviously Trichogrammatoidea cryptophobia, but there's some other additional egg parasitoids that aren't commercially available, and then various different predators as well will actually attack those eggs. What you can do is, I know there's a lot of people working in the rose industry, is do some training on how to scout for eggs. Typically, when the moths lay eggs, they will tend to lay in the same sort of area. For macadamias, it's in the suture of the macadamia. For citrus, it's towards the calyx end or towards the navel end. And in roses, the brief research that I did, it seems like it's on the lower bud area, but I can't say that with confidence. It's just what I've looked into. So once you start to understand, where are they laying the eggs, and once you start to get your eye in on how to identify them, you can scout for them quite successfully, but it is difficult. To come back to the mass trapping, so for the mass trapping, it seems like in peppers, they do use it, as well as in the avocados, and about 90 to 120 traps per hectare for the mass trapping.
Thank you. Sorry, we're almost running out of time, but Mesfin is asked it again. What is the ratio, the sex ratio of FCM, male to female?
Yeah. So I can talk about this in our cropping systems in citrus and table grapes. And typically it's between a 50-50 to a 1-to-2 male to female ratio. I'm not 100% sure in roses, different types of crops can affect that ratio, but typically it's not a huge difference between the numbers of males to females for this species. It's about 50-50 to 1:2.
Good.
Something I just wanted to point out is obviously the monitoring tells us about the population of the pest from the male perspective, but a lot of work has been done to show that it doesn't correlate directly to egg lay or infestation. So the monitoring is really used as an indication of when your population is increasing so that you can make those management decisions. But if you get like 5 males in a trap, it doesn't mean you're going to get X number of females or infestation. It's very difficult to make those correlations.
Thank you. There's a question on the quality of the mating disruptor pheromone.
Yes.
So there's Mr. Pradeep. He's mentioning that they used the mating disruptor, but it was catching other species, and
Yes.
very few FCM. If you could comment on that as well.
So obviously the mating disruption, the mating disruption, really what mating disruption is different types of dispensers that can dispense pheromone, right? So you can get any type of pheromone that you can make into a synthetic product that is able to release volatiles can be put into mating disruption products or dispensers. So you do get other products for other species, but the FCM one is targeted towards that FCM to the FCM males, and the reason being is it's the specific blend of those pheromones that are only released by the females of the species.
Yeah, so maybe the question is not quite clear actually, because mating disruption, you're not supposed to be catching anything unless you also have the delta traps for monitoring. Yeah. Maybe we'll get through to Pradeep just to clarify the same.
Yeah, sorry, maybe I misunderstood what he was asking.
And then somebody also is mentioning that the use of repellents in the management of moths. Any experience with that? So like garlic extracts.
Yeah, I do.
He's actually mentioning that a product that is using NemGuard and is getting very good results apparently.
Yeah, I do have a little bit of experience using various other volatiles and repellents, etc. They can be useful for moths and they can be employed quite successfully. I mean, I know in Kenya, they've got quite successful use of the push-pull systems. I don't think it has been as well researched for FCM. There's a couple of people doing research here in South Africa on various repellents that can be used, but you don't always see that efficacy on a wider field scale. However, I do think for roses, especially roses in greenhouses, that it can likely be used more effectively because the scales aren't as broad and you're not having as many environmental weather effects, etc. So I have also heard from people that their neem guard is working quite well and the garlic extract is working quite well. But specifically, I haven't researched that. And so it's more observational than anything else. But it does, repellents do work for other moth species. So it is possible that it's working as a repellent.
Thank you.
Yeah, sorry.
Then this...
I'm sure the answer's a little vague.
Sure. Yeah, I think we are running out of time. Maybe 5 more minutes. We stop at 12:10 so that we don't stretch this too far. But maybe a couple of more. Would mass trapping as a loan management measure be effective in dealing with FCM? That's a question from Mafeni.
No, like I said in the talk, there is no loan method that would be effective. So I do not recommend that, especially some of these softer methods like mating disruption, like mass trapping, that they be used completely alone. You can use them alone for up to a period of time, but I would still do the monitoring and then use your foliar applications as necessary when infestations or moth peaks are present. The reason being is, if we were just looking at a purely economic threshold, then you could potentially use things by themselves. But we're not only looking at an economic threshold, we're looking at a phytosanitary threshold. And phytosanitary thresholds are very low, like I said, almost a zero tolerance. And to get that zero tolerance, it's almost impossible unless you do an integrated method. So whether your integration is, cultural practices or whether it is insecticidal practices, etc., you always have to do other methods. And especially for me, the sanitation is one of the number one methods that you can do. And, for sure, that's not a commercial recommendation or going to benefit anybody from a commercial sense, but it's one of the best things that you can do. You can reduce your infestation by up to 70% with good cultural practices, and then you layer your other practices over that. So mass trapping is another practice that you can layer over that, and then I would still try to see, are we getting any increases throughout the season, and then do an appropriate foliar application.
Thank you, Megan. This one for Dr. Mary. You mentioned something on screening, including the top vents. There's a question from Mohammed, how effective this is. If you got some experience with that, I've got a farmer whom I know has screened his greenhouses, and he testifies that there's no problem with FCM so far. I know you guys are working on something on that. If you could shed some more light on that, Dr. Mary, kindly.
Okay, thank you, John. This is something that we are experiencing for the first time, because initially people were free to open their curtains whenever they felt like to take care of humidity. But we have seen, since the time we installed the side netting, or we implemented the aspect of side netting, we have seen a lot of improvement in terms of the pest management. That means when they do the monitoring traps on the outside and the inside, they have realized that the population that is outside is usually higher than what is inside, and therefore the risk has been minimized. Companies themselves have indicated that they have seen a great improvement in terms of pest entry into the greenhouse, and therefore I can say the screening has been quite important. For the top vent, I think this one, I have to say that it depends on where you are found, because for people who are found in very high altitude areas, you realize the temperatures are quite low, and therefore if you are to cover up the vents, it could have some adverse effect on quality. But for some, in a number of companies, particularly people who are in slopey areas where the pest can be propelled by the wind, it has been quite helpful in making sure that the pest does not get in. So the top vent, it can be used carefully depending on where you are, the altitude where you are found, and also the terrain, which of the greenhouses are situation. Thank you.
Thank you. And actually, as we are talking, I didn't want to mention whom, but he's come out to identify himself. Stefano Manella. he actually confirms that monitoring traps in the greenhouses with insect-proof nets are empty, no FCM, so it actually works. But of course, I think as Dr. Mary has already mentioned, of course, there'd be issues with the environmental control, given the other fungal infections and things that will have to be taken care of. Thank you, Stefano, for that. Well noted. Ladies and gentlemen, I think we'd not want to stretch this beyond. I promise that we'll try to get in touch with each and everyone who's put up a question on the Q&A that we've not responded to. And mine is to say thank you so much for making time to join us for this webinar. We promise to do more and more of this, as we always believe that knowledge is key. And in pest management, of course, the foundation of any successful pest management begins with a thorough understanding of the biology. So I believe as far as FCM is concerned, we've gotten something that we can put into action to help us in mitigating this thorn in our flesh currently. Thank you very much. I think we'd like to stop at that particular moment. And not forgetting to say a special thank you to our partners, KEFIS, Dr. Mary, we really appreciate your time. Very special thank you also to River Bioscience. Dr. Megan, thank you so much for making time for this. We hope to invite you for more and more. Thank you very much.
Before we finish, John, if you wouldn't mind just sharing the questions in the group chat with me, because I can't access them, so I can, maybe put together like an email answering some of those questions in case we didn't get everybody. Yeah.
Thank you. Lewis, if you could kindly take care of that. Thanks.
Yeah.
Bye-bye, everybody. Thank you so much.
Bye, bye everyone.
Bye, everyone.
Bye. Thank you.
Bioline Agro Sciences, cultivating bio-alliances.